In this video we will discuss about O-Glycosylation. It is the post-translation modification that occurs in the mammalian cells. So apart from the O-Glycosylation, this N-glycosylation will also occur, but in this video our focus will be on O-Glycosylation. So we will start with this TN antigen, which is the first monosaccharide that is attached to the serine and threonine, and O-Glycosylation mainly occurs at serine and threonine part using this uh, transferase, that is PP galNAc-T. It will add galNAc to the serine and threonine, which will cause this TN antigens, and from this TN antigens it will either form this core 1 to core 4 structures or the silyl TL antigens. In order to form the silyl TL antigens, this ST6 galNAc transferase is needed, and all these O-glycosylations occurs mainly in the Golgi body. So from this TN antigens that is shown in the middle figure, is with the core 3 galNAc-T, the core 3 GlycNAc-T, the GlycNAc is attached through this beta 1,3 positions, which is the core 3 structures, and through this C2 GN T2, another GlycNAc is attached to the galNAc at beta 1,6, and this is the core 4 structures. And through the core 1 GalT1 or C1 GalT1 or the C1 GalT, the core 1 structures are formed, which is this galactose is attached to the galNAc using beta 1,3 linkage, and from that and this is usually known as T antigens, and from this C2 GNT GlycNAc will be attached to the beta 1,6 position, and this is, you can say that, core 2 extensions. And if the GlycNAc-T is attached through the in which case GlycNAc is attached to the galactose, then it is the core 1 extensions, because we can, this kind of structures, these core 2 structures contains this galactose and the GlycNAc attached to the galNAc, whereas in the core 1 structures galactose is attached to the galNAc and the GlycNAc is attached to the galactose. This is, you can say, the core 1 extension. This is the core 2 extensions that will carry forward. And if the ST3 GAL1 is act on the core 1 structures, then in that case a sialic acid will be attached to the galactose, and if the ST6 galNAc is attached, then GlycNAc is attached to the galNAc at beta 1,6 alpha 1,6 linkage. So these are some of the O-glycosylations that occurs, and in the terminal GlycNAc residues that is attached, terminal GlycNAc residues, in which case the GlycNAc residues are attached to the serine and threonine instead of galNAc. So in that case, when the GlycNAc is attached, then this beta 1,4 beta 4 GalT is act, then it will cause this galactose attached to the GlycNAc, and the ST3 GAL1 will cause sialic acid attached to the GlycNAc, and after that this alpha 3 FUT T will cause the fucose attached to the alpha 3 positions of the this core GlycNAc. So these structures are commonly known as silyl lysases, and these are usually overexpressed in the cancer cells. Similarly, if the beta 3 GalT act, then it will attach galactose but at beta 1,3 positions, and after that the process is same. The ST3 gal will attach sialic acid, and the last one also change the position. This alpha 4 T will attach fucose at alpha 1,4 positions, which is the silyl lysase position. And these are also overexpressed in the cancer cells, mainly in the pancreatic cancer cells, because of the dysregulations in the O-glycosylations pathway. And finally, where the mainly where are this O-Glycosylation occurs mainly in the within the Golgi body. So in the here the from the ER the from the ER the protein transports to the Golgi, and in the Golgi part, what happens is that this GalNAc and this GalNAc kind of transferase are mainly present in this Golgi body part, or we can say that this core 1 GalT1 and PP galactase are mainly present. So here the O-glycosylation begins. Galnac is the monosaccharide that I have spell is the enzymes that are present in the Golgi. Golgi is core 1 GalT1 mainly and PP galactase, and some of the ST6 galnac also and ST3 gal1, but mainly the PP galactase and core 1 GalT1. And in the medial one, the enzymes that are mainly present are ST3 gal1 now, and B4 gal-T and ST6 galnac, and also to some extent the core 1 gal-T and PP galnac-T. And in the top one, the amount of ST6 galnac increases and this PP galnac-T decreases, and the ST3 gal also increases, and and along with that B4 gal-T also increases. So we can see that PP galnac-T amount decreases, and also the other extensions related glycotransferase increases as we approaches towards uh, trans-Golgi network. So this is some of the basic overview about O-glycosylation. If you need to understand more about the O-glycosylation in detail, you can go through the reference that has been provided in the description. Thank you for your time.